Thanks very much, Dana, and I'm very happy to be here. I love coming here a number of times. We were discussing what meeting when Rod and I were at the same meeting, but he left and I came and we didn't even know we were at the same meeting. So I love coming here. So have another meeting with you.
Wendy is a person that don't matter what happened when you invite her to come, she comes. She never does wrong.
So today I'm going to talk about one of the projects in the lab. We have a couple. I can talk to you about some of our MS multiple sclerosis antibody projects, but I'm talk about this, which I've been talking about a little bit over the years, where I could never get any funding to support this, and it was actually a really intriguing project, and suddenly it clicked. And so we have some. I'm going to show you a bunch of interesting data on how this system has been working. So, talking about axonal signaling regulating oligo differentiation. So as Mark, Janine has said, we've been studying myelination for years, and a lot of the work was really focused on mTOR signaling. So we've had many papers since for almost 20 years about how AKT and mTOR drive molecular differentiation and myelination in oligodendrocytes. And so as we were doing those studies, there were a lot of papers that were coming out that were suggesting that mTOR signaling in neurons was also incredibly important. And if you knocked out Raptor or Richter in pretty much all neural progenitor cells, you were changing cortical neurons in many different ways, and you were changing the morphology and a variety of different deficits inside the skeleton. So it was clear that we were looking at mTOR only in oligodendrocytes, we thought, but things that we were doing could be affecting what's happening in neurons. And so we then realized if we looked at just normal tissue, there's an incredible amount, far more mTOR signaling in neurons than in oligodendrocytes. And when we treat it with rapamycin, that doesn't work. It doesn't work here. It's the wrong kind of screen, but whatever. So that when we hit with rapamycin, we could knock out all the neuronal signaling of mTOR. But also, what we were trying to look at was the oligodendrocytes. But when we did this, next slide, we also noticed that suddenly we had this increased oligodendrocyte differentiation. So we were hitting mTOR signaling far more in neurons than in oligos, and actually in this context, it was actually inducing differentiation. It was like, this is not making a lot of sense. So we decided to go with the next CRE, which is a gene that Klaus Nave identified a long time ago, that regulates recombination in post-mitotic cortical neurons. And so we were able to try to look at what was going to happen if you knocked out different things in cortical neurons, and what did that do to oligodendrocytes. So next. And what we did this, so we did it with either Raptor or Richter, which is mTOR1 or mTOR2, and the Raptor impact is very, very strong. So we started looking mostly at the Richter context, and you see very early, very major developmental changes in the axonal development in these next CRE Richter floxed mice by P8. It does recover as the animals get older, but you have dramatic changes in corpus callosum axons. And you see quickly, you see changes in the way the oligodendrocyte progenitor cells look. They are already at this P8 time point, changed their shoal analysis. There's the number of intersections is definitely changed as a consequence of this change in the way the neurons are developing. And we did see an increase in the SOX2 population there on the left, which is an early transcription factor in the whole oligo lineage. Proliferation, we didn't see much change in the OLIG2 population, very little difference in the way that the cells were proliferating. So it didn't look as if we were getting more SOX2 positive cells because they were proliferating more, but perhaps that they were not a lot, they were changed in the way they were differentiated. So we also then saw that there was this increase, as we had seen with the rapamycin experiment, there's an increase in expression of ENPP6 positive cells. That's a transcriptional factor that was identified about 10 years ago that is on in the pre-myelinating cells, not in the differentiated, not the fully myelinated cells. But we saw this increase in the oligodendrocyte lineage cells in the differentiated state. And many of them went on to die. So there were a very high number of tunnel positive cells in that context. So increased ENPP6 cells and increased cell death. Presumably because they're not getting, if you saw the image of the axons, they're not getting the normal axonal targets that would be appropriate for a pre-myelinated cell, so they die. So the question really was, what's happening and how are they being altered because there's altered axons in that corpus callosum. We clearly see Richter loss in those neurons having a dramatic effect on neuron axon development. And as multiple study sections pointed out to you, that is a far too complex system because you have the loss, many changes as a consequence of the Richter and mTORC2 loss. And so this was, we were trying to figure out better ways to look at this question in the developmental context because we just couldn't really get clean answers out of this Richter flux channel. So clearly we do know from this that during early brain development, in the absence of appropriate axons, they do prematurely differentiate, and they also will then die. But one of the questions really was, can we separate out just simple neuronal activity relative to all the other things that are happening when you have this major skeletal change in the development of the axons? So we then went to look at that question. And when you look at the literature, literally for the last 10 years, there's an absolute dogma that increased neuronal activity increases oligodendrocyte differentiation and myelination. And presuming in other contexts that reduced neuronal activity reduces oligodendrocyte differentiation. So this was out of Michelle Manji's paper on looking at this question with development. And a variety of other papers came out in that same time frame from a variety of different labs showing that the learning process of motor learning was required new CNS myelination for the learning to occur. So increased activity on the running wheel was inducing increased myelination, and if you prevented that, you didn't get learning. So learning was part of, required some, in this motor learning context, required some degree of new myelination in that region. Out of Bruce Appel's lab, neuronal activity was showing in the zebrafish that you have a bias on which axons get myelinated, active ones getting myelinated, inactive ones getting less myelinated. And then out of Dave Lyon's lab, he showed that synaptic vesicles are required to be released from the neurons as they're developing again in the zebrafish to regulate the amount of the number of myelin sheaths that are happening in the developing spinal cord in the zebrafish. So what are these interactions that are happening between neurons and oligodendrocytes when these axons are still developing? Does activity always drive oligodendrogenesis and myelination? So, okay, so there were some papers out there. So there's tons of papers that say increased neuronal activity increases myelination. There were some hints that there might be some other kinds of regulation. So there was a paper that showed that the whisker plucking in early brain development in mice increased oligoprogenitor cell proliferation and differentiation in the deprived area of the ventral cortex. And then that if you decrease glutamatergic neuronal activity, both in vivo and in slices, you can increase OPC proliferation and oligodifferentiation. So there were hints that some aspect of decreased neuronal activity might actually induce something with differentiation of oligodendrocytes. And so we developed the hypothesis that these neuronal activities are inhibiting oligodifferentiation in the early development before the axons are mature enough to get myelinated, which makes sense in some reasonable way, and that is actually something we could investigate. So the best way that we could figure out to separate these morphological changes that we were seeing in the Richter knockout animals from simple electrical activity was to go after dreads. These are artificially created receptors that should only respond to certain things like CNO. And you can overexpress that in a particular cell type and then see what that does to the activity. So Tessa Allen is a graduate student in my lab. She's really driven these projects with the whole dread work. And we collaborated with Wan-Chan Oh in our pharmacology department, who is a physiologist, which we need to be able to investigate what dreads are doing in this context. So we knew that developing axons are actually making synapses on oligoprogenitor cells during development, and that this is probably going through AMPA receptors, because if you treat with NBQX, even at P7, so very early in development, there are these AMPA-mediated currents in the oligodendrocyte progenitors that are sensitive to NBQX, and that continues on through the development. So the intent was then to use threads to selectively alter activity in cortical neurons in the early postnatal CNS development. And so Wan-Chan had been also looking at this question. He doesn't care about oligodendrocytes, although he's very intrigued with these synapses on them. But his whole focus has been on serotonergic cortical neurons and their impact on a variety of different elements. And so this paper was a paper from last year where they used the GI dread, which is the inhibitory dread, to show that those serotonergic cortical neurons had an impact on all sorts of different things, with reduced spine density and physiology. So he's been helping us to look at how does altering cortical neuronal activity during early postnatal development regulate the impact of developmental oligogenesis. So what Tessa did was to inject either the GI or the GQ dread into the P0 mouse. So she takes it from an entire next Cree population. So 50% of those animals are going to be Cree and 50% will be negative. So she'd get half the animals will have been overexpressing dread in the next Cree cortical neurons. And then she's looking at the development across the corpus callosum in that developmental period. She's in every day twice to feed these animals, and she's worked out this beautiful way of feeding the animals CNO, feeding them literally with CNO twice a day to look at really good recombination and then activity impact with the CNO as a consequence of expression of the dreads in these cortical neurons. And when she does that, then she takes the tissue at P8 and then looks at all these different markers, primarily focused on the PDGF as the marker for the progenitor cells, BCAS1, which has been characterized to be predominantly up and then down in premyelinating oligos, and then CC1 and other late markers for the mature oligos. And so we are in the midst of doing some physiology on these animals with Wan-Chan, but in the meantime, she has looked at whether their C-FOS expression is downregulated in these GI, inhibitory dread animals. And if you look at the upper, the lower, at the higher mag, you see the C-FOS is on in the control tissue. It's reduced significantly in the next Cree animals, where the dread at the lower bottom of this thing is expressed, whereas it's not expressed in control animals. So yes, we do see reduced C-FOS. She's just started the physiology with Wan-Chan to look at does that, and in an initial analysis, yes, it does clearly have an impact on the electrical activity of those neurons using patch clamp. So what does this do? We'll go back.
please.
So,
Sorry.
she's inhibiting neuronal activity with GI dread, and she's
This-
increasing just as with the earlier studies. She does see an increase in the BCAS1, so that's pre-myelinating cells in that context. She had now looked at corpus callosum and in cortex. So oligonucleotide differentiation in both regions, and these are neurons that are projecting through cortex and over the corpus callosum, are affected by knocking down neuronal activity in these cortical neurons. It doesn't seem to affect, in this context, it doesn't seem to affect proliferation of the progenitor cells. So they're being pushed to differentiate, but there's no obvious increase in proliferation of these cells. Thanks. So we know now that in early development, decreased neuronal activity is increasing differentiation. In this context, we don't see any change in the dying progenitor cell, pre-myelinating cell. So at least in this early time frame. So it's probably because the neurons, the axons are actually in pretty good shape. They're just not very active in this context. So then she went to look at, well, what happens if you increase neuronal activity as opposed to decrease? And there we now have an increase in the numbers of OPCs, progenitor cells, in that context, primarily in both cortex and corpus callosum. So we see an increase in the progenitor cells. And she then wanted to see whether this was because there was increased proliferation, or was it a change in the differentiation? And yes, at least in corpus callosum, and to some extent, she did EDU experiments to look at proliferation of these cells, and then the next one. And there we see, at least in the corpus callosum, we do see an increase in proliferation of the oligonucleotide progenitor cells. Didn't see anything like that in the cortex. But so this is increasing the neuronal activity, increasing proliferation of progenitor cells, and then it's decreasing differentiation. So it's the opposite. Trying to keep this straight in our heads is always hard. Increased activity of the neurons is decreasing differentiation. Decreased activity of the neurons is increasing differentiation. So in this context where she had increased neuronal activity with a GQ dread, she's got decreased differentiation in both corpus callosum and cortex. So from all this, we now know that decreased neuronal activity increases differentiation, whereas increased neuronal stimulates some proliferation of OPCs and definitely inhibits OPC differentiation. Okay, so whereas in mature animals, neuronal activity is promoting oligogenesis, we have at least 2 models now in which altered neuronal development has the opposite effect on all of the differentiation. So early in development, decreased neuronal activity increases differentiation, whereas increased neuronal activity inhibits the differentiation. And so, and this is all Tessa's idea. She wanted to see, well, was there a more physiologic way to look at the same question? And so she went on to look at, next slide, the olfactory system. And the olfactory system is a very nice model for all sorts of different reasons, sensory evoked neuronal activity. And so the olfactory bulbs are projecting to cortex, and they are responding to odors as the animal develops. And so all of the neuronal development in that context is based on breathing and odor, taking in odors, which then are hitting the glomeruli in the olfactory bulb. The mitral cells and the tufted cells then project through the lateral olfactory tract to the cortex. So there's data that if you do unilateral neurosecretion, so you're only blocking one nostril, that you get a significant reduction, we've known this for 150 years, of olfactory input in that one nostril. So the olfactory input for one nostril into the olfactory bulb is altered. And so that has been shown in the last 10, 15 years, that that's also changing myelination in that context. Generally looking at adult mice, so you do the unilateral olfactory occlusion, and you see changes, and most of the people have been looking at adult. So she had to sort of dig through how best to look at this question during early development. And so if you have neurous occlusion, you have reduced myelination in the adult. So now what she's doing is doing the same unilateral neurous occlusion at P0 and studying the immunohistochemistry of the oligodendrocytes, and then looking at what's happening in general in later time points. So she's looking in the lateral olfactory tract. And she's got one animal that she's got these data on that. So one animal about the CFOS. And the pain in the neck is that mitral cells, many neurons in the olfactory bulb express nu-N. Of course, the mitral cells are the ones that do not. So she can pick those, no, go back. She can pick those up, go back.
But
Yeah, there.
this.
So she can pick the mitral cells up because they are Nissl positive, nu-N negative. And she has in one animal shown that the non-occluded FOS positive, Nissl positive, nu-N positive cells are greater than in the occluded side. So she's got a whole series of animals that she's going to look at, but at least this is consistent with the idea that she is, and the data, clearly that she has reduced neuronal activity as a consequence of this. And so then when she looks at, in that olfactory tract domain, at either progenitor cells with the PDGF or BCAS cells as looking at the pre-myelinating cells, there doesn't seem to be any major change in the non-included versus the occluded sides for the numbers of progenitor cells, the PDGF positive cells. But again, in every animal, she's comparing each line here is the same animal. It's looking at males and females. So she's seeing in every animal, she's seeing a change to an increase on the occluded side of the numbers of differentiating oligodendrocytes. So again, in this model, oh, thanks. Again, reducing the neuronal activity in this context is increasing the differentiation. So overall, we now have 3 model systems showing that when you reduce neuronal activity during early development, you're getting an increased differentiation. The Richter mice have this change, but those pre-myelinating cells die. In the DREAD context, the neurons are relatively normal, but relatively clearly reduced neuronal activity. We again get this, and we can actually look at the increase in activity in that context, and we get the opposite. And then now in the olfactory system, she's also seeing that, presuming we can show better numbers of animals with the CFOS loss, then again, if you have that reduction, you have a premature differentiation. So we finally got some money to study this. And so she's really pursuing this question. The next slide. And the next. Okay, so now what's the mechanism we have? We know, we think the logic is, that you don't want to be trying to differentiate all the dendrocytes when the neurons are not, the axons are not appropriate. But what is the regulation of this? And so we've been going after gene expression. And so what she's been doing is doing single cell analyses. So if you do the GI DREAD, the inhibitory DREAD, and then you're doing the CNO from P2 to P8, and then you do single cell analysis. So she isolates PDGF positive, O4 positive cells, and then does single cell analysis. Yes. And so we can see a UMAP analysis on a lot of different clusters. And this is done in collaboration with a bioinformatics person that is a long-term collaborator. And so when you start to characterize those, you see that the progenitor population is this group on the right, which is all PDGF receptor positive, and then the next group is the next slide. So you can define these groups into OPCs and other cells, and then next slide. The other cells include BCAS positive, differentiating oligodendrocytes. So we have a pretty decent split between the OPC clusters and the and then later stage differentiation. Next slide. And so we can go through a whole series of different things. So TCF7L2, which I'll come back to, is clearly upregulated in the GI DREAD animal cells and pushing them. ENPP6, which I said is also a differentiation marker for the premyelinating cells. And interesting, GPR17, which has been a very interesting gene looked at by a number of labs that has to be expressed, but is doesn't stay expressed, and so that's also going up in this population. And then the SOX2 population, which we had also seen increased in the next pre-animals, is also increased in this population, but it's also up in the progenitor population as well as the differentiated cells. So we have a whole series of genes, and when you run it through a variety of different pathways, you've seen changes in all sorts of different things that could be regulatory in early stage development of these cells. One of the genes that's high, that stays high in the progenitor population, is EGR1, which is FROX24. So what that is doing to change the differentiation, change the state of the progenitor cells, is something that we're kind of interested in characterizing. Okay, so we know clearly OPCs have a very strong innate differentiation drive, and that during early development, neural activity is inhibiting the transition to differentiated to premyelinating cells. And so we've been focusing in a little bit on the SOX2 question as a timer of OPC differentiation. We think that could be a driver in this context. But newer data would suggest there's other areas that we need to focus on. But we really want to know what is the signaling system that's blocking premature differentiation. So we can do pseudo-time analysis in these cells, and we can look at differentiation over time with the progenitor population moving into the more mature population, and so it's a proxy pseudo-time analysis, or it's a proxy for changes in developmental time. And so if you put all the cell populations together, you have the progenitor population, the premyelinating BCAS population, and the more mature MVP positive. So 0 in pseudo-time would be this progenitor population, 1 would be the differentiating cells, and 2 would be the late stage mature cells. And so when you look at this with the pre-positive versus the pre-negative population, you're seeing a loss in the pre-positive animals of the progenitor population, and then an increase in the overall differentiation state, numbers of cells in the differentiation state of the oligolineage. And what Tessa's focused in on has been the AMPA receptors, and she's seeing definite changes with increases in several of the AMPA receptors. And so she's really going after some questions in terms of trying to manipulate AMPA receptor function and see what that's doing to the differentiation of the oligodendrocytes. We've now gone, and this is the most recent data we've got, we've now gone to really characterize, we did the single cell on the inhibited set where we see increased differentiation in 14 different contexts. And we can compare that directly to the population that is with the GQ dread that has the reduced differentiation in the oligodendrocytes. So we're now in the midst of trying to do some analyses comparing the P8 GI dread animals, cells from the GI treated animals to the GQ treated animals. And so what we are looking at, this is years later analysis, so some of the UMAP assessments are different, but we definitely see clustering in the same kind of context with OPCs and pre-myelinating oligos and then early myelinating oligos in these populations. And so what we're now doing is comparing the data that we've got from the GI dread to the GQ dread, looking at using all of the wild type animals from both experiments to compare what's going on in the wild type relative to the GQ or the GI. And this is all done with my collaborator Ken Jones, who knows this, is teaching me at this. So what I'm going to tell you is stuff that I'm just learning. So what he's done is taking this into IPA to look at the protein relationships in terms of what's driving this system. And the analysis through IPA of the proteins and the activation state of the proteins gives you all these different kinds of data. So if there's a really low activation state for the protein, that's in the blue, and if it's a very high activation state, then that's in the red. And for me, the only way I can understand this is if I look at the cluster 3 P8GI dread, so the fourth column over there. I see a lot of those genes. So these genes all popped up out of this analysis. And there's a high activation state in the GI context, which is the context where the cells are differentiating prematurely. So all of these genes seem to be going up in cluster 3, which is a pre-myelinating cell population in the GI context. That same generally group of proteins in the GQ context is really coming up in the second line, the cluster 0, which is one of the early stage progenitor populations. So out of all of this, which is TCF7L2, which is a transcription factor, SOX2, which is a transcription factor, contactin B, YAP1, and TGF-beta seem to be linked in terms of the changes that are happening when they're being pushed to differentiate prematurely or being inhibited from premature differentiation. And so what if to take just TCF7L2, it's turning out to be, if you just look across the different clusters, so 1 and 0 being the progenitor populations, 3, 2, and 7, which isn't even on there, are the late stage differentiation states. So if you look at, in the next slide, if you take the cluster 0 analysis from the GI treated animals, TCF7 is not in the GI context. It's lower than it is in the cluster 0. This is progenitor population in the GQ, which is being prevented from differentiation. And if you look at the next one, which is looking also at TCF7, this is cluster 3, is looking like it's had a significant, this is the Z-score of over 2 is the first thing that Ken saw and said, this is really significant. But the TCF7 Z-score of over 2 in the GI dread animals relative to a below -2 in the GQ animals, where oligoprogenitor population differentiation is reduced, is extremely significant. And so out of all of this, he's gone on to come up with this string DB analysis, which is really talking about activation states of proteins in these contexts. And all these little lines are different things in the literature from different ways of analysis, whether it's co-expression, they're showing up in the same paper, they're expressed from something that's coordinating their common regulation. So all of these lines say something different about what the relationship is amongst these. But his interpretation of these data is that TCF7L2, which is a transcription factor that only comes up in the progenitors as they differentiate, is a driver, and it's not just gene expression, but that these protein relationships, activation states and inactivation states, are creating this pathway that is contactin and YAP1 and SOX2 and TGF-beta regulating aspects of how the progenitor population, which is the PDGF receptor A population, then moves on into all of these different protein interactions of MYRF, which is a myelin oligocyte specific driver of differentiation, and all these different myelin proteins. So this is where we are. This is what we are now going to have to dig into these data and really figure out what is really driving this. And is TCF7L2 really a major driver in the system? And if so, how does it interact with contactin and YAP and this whole pathway to drive or inhibit differentiation along genocytes? And the one last thing I'll point out is the next slide. Oh, so these are just what I've told you 14 times so far, that we know that these things are being regulated by neuronal activity. You can manipulate it by changing neuronal activity, and that we have a potential gene protein regulatory pathway that we can now investigate in terms of regulating the timing of this OPC differentiation. And one interesting thing that we are now in the midst of doing, we've got these GQ data from the P2 to P8 timeframe. We know we have the gene expression data from that, where we know oligoprogenitor proliferation is increased and differentiation is decreased. And Ben Emery did the same experiment 10 years ago, well, 6 years ago, whatever, where they did a similar GQ red experiment, but instead of treating this really early time frame, they treated much later, which is the typical neuronal activity increases differentiation. So they were treating the CNO from P19 to P25, and in that context, OPC proliferation was increased, differentiation was increased, and there was an increased number of axons that had higher probability of being myelinated, and myelin is thicker. So what's happening in terms of gene expression in these early time points relative to the older time points? So we have the GQ data already, and what she's now trying to do is generate a series of animals that she can go and do the single cell analysis out of with the older ones, and really begin to look at what gene expression changes in the control population. I mean, there's obviously something different about those OPCs in the early stage relative to the older stage. And then, of course, with the dread expressions, how have those changes hit the things so that they change entirely what the oligonucleotide does. So, lastly, this, as I mentioned, I'll drink water in a minute. Tessa has been really driving this thing for the last 3 years. She's a graduate student in the lab. Was telling me her fellowship got an incredible score, and we are not yet funded for it because craziness in the United States. Graham Pete is also he's doing some amazing stuff with bioinformatics. He's actually got a machine learning thing about multiple sclerosis antibodies. And he's been helping test it. So they were both graduate students in the lab. Katie Bercury was the first person to really identify this characterization of the neuronal changes in the Richter animals. Bella, some of you may know Bella, who was absolutely wonderful. She's now in Italy. She's been working on that Richter story, and we just never got it finished before she had to leave. And Jennifer Bourne does all the EM work we do. Wan Chan is, again, this physiologist who is really wonderful. Ken, also the bioinformatics, and then all of our mTOR stuff started out of this incredible collaboration with Terry Wood at Rutgers and my lab. Thank you.
Thank you all very much, Wendy, for your wonderful talk and for this different point of view of oligodendroglial differentiation through neuron activation. And your presentation with Jorge, we used to say that you are one of the 5 golden stars in the Mayan information. Your talk is really confirmed our dream. So, talk is open.
No, del journal no veo lo que es. Pero no había otro micrófono, dijeron que iban a poner otra cosa. No hay otra.
Really nice data. It's really great that you follow an observation, all down to molecular points. Really, really good. In thinking about mechanism, I wondered the Notch ligand signaling described first by Ben Harris some years back.
Yeah.
How crucial that is.
So it didn't come up in this last analysis, the Notch pathway, but when Ken was doing the initial analysis, it was definitely changed, as is the SOX2 story. So it may be that we may need to dig into that just in terms of the increased differentiation question. Where does the Notch change and how does it? But we don't have any answers to that.
One more question to follow up. In thinking about time points, early time point versus a later time point, looking at intrinsic changes, you might also have the extrinsic.
I'm sure it is. The extracellular matrix, also any number of, and the neurons. And so clearly, we can get after gene expression in those neurons very straight. We're using the NextCree through all the system. So the problem with looking at those kinds of changes is that they're so massive. You've got the brain development and all the neuronal connections and all those changes. So we're focused initially on what are the gene expression changes that are fundamental in the wild type context between P8 and P25, but also than what the red things do, because it's an easier population to really, the differentiation of that population is so well characterized that it's easier to interpret. So that's, but yeah, the extracellular matrix, the neuronal function are totally different at P8 versus P25, you know, and so yeah, hopefully we have a lot of interesting work. Did you have a question? Yeah.
Very basic question. Yeah, regarding the what's the extension of the effect of the all these proliferation? Like how close to the axon you have this effect of inhibition of these
Yeah, I mean, it's easier to do that in the cortex because you can really see the cortex goes in the body, so it's much harder to see proliferation. She hasn't really looked at that. You're talking about whether the progenitor, proliferation.
You're thinking of the so factor release by axon
Sure.
by activation.
Yeah.
Well, then you have limitation more probably in the
Distance.
yeah, with the distance more probably
Yeah.
So I was thinking, the other question is how can you rule out also a migration effect?
No, not with.
Because you know that Mac and Logo A both complement on migration of the that was the model that were what and I think that has a migration delay. So you're not going to get this effect on the progenitor, but at the same time, then fully migrating like Mac for sure, no way, and the combination really
change the interactions.
Something that you really confirm that it's a releasing factor. If so, then I think that's very good. The AMPA receptor glutamate effect, makes sense.
Yeah. Well, this is why. So she's now Tessa. It's now it's very hard to do those experiments in vivo.
Yeah.
But she now is getting cortical slice explants, and I think because her focus right now is really her hypothesis for the thesis is that the AMPA receptor signaling is changing in consequence of these changes. So it's much harder to do that in a vivo context. She's got this. And she now has a bunch of slice experiments going that look like she will able, first of all, the differentiation context changes. So in the slice mode, so she's letting them go a long time and everything. So we're now trying to figure out what the right time frame is to try to do those experiments. And see if that's where she can look at knowledge and look at a variety of things, morphology of cells, morphology of neurons. So yeah, so right now I think that's where we can go.
But if this glutamate, or something related, you may explain also why you have increased myelination or tumor myelination and then not if you do the integration with your own.
Yeah.
Because
They may be completely changed.
We probably will go because the story with the reported MSC and Cipina and glutamate interchange, which basically you come up with a lot of cells, GSH basically. It makes sense that that's why I'm wondering which one is the extent of the action of the effect on the surrounding. And that's going to change the accelerometer magically make, but I think it's not more to the vision itself.
Well, and I mean, the whole back to the experiments, the collection of activity and the release of some active vesicles from developing axons is driving some aspect of the so for developments. So yeah, I think so in the mouse context, I think we really do need to be using some kind of excellent to really dissect those things. That's what we're going to do. Another question?
No question. When you block the olfactory nerve, I guess, which kind of, there is a kind of special neuron that is participating in the phenomena of.
So the olfactory system, there's a lot of receptors in the nasal domain. those project up to the olfactory bulb.
Mhm.
So they're all, it's all a glutamatergic system, that leads to the early development. So those are projecting, and those are obviously being blocked. But what she's measuring is the projections out of the olfactory bulb to the other parts of the cortex. And those are all, again, glutamatergic projections.
The trigeminal nerve have something to do in this mechanism, which is trigeminal.
I don't think so. These are-
It's
I mean, these are
only
all,
olfactory nerve.
yeah, it's all, this is projections related out of the olfactory bulb through the microcell projections to the olfactory cortex. Yeah.
Oh, I enjoy really very much, very nice, very interesting point of view of legal differentiation. More questions? Yes, Vanessa. She's Vanessa Maclin. What, can you use that? There is another microphone.
This.
Well, thank you for the great presentation.
Sure.
This is a very simple question. I mean, maybe you mentioned that I was wondering how you perform it in a kind of different.
It's cauterizing.
Not out loud,
No, she wants to do that actually, but it's far more fun if you're doing. She wants to do the pause because you can then undo that.
Yeah.
You can't undo the cauterization. So yeah. So right now she's just doing the straight analysis. But it would be interesting then, at what point could you start having neuronal activity again to change it? So and when you think about it, the whole. No, I take that back. But the cauterizations that were published were like P15 time point. She's doing it at P0. So plugging a P0 up would be very hard.
Yeah. And you see a nipsey?
When you analyze the effect of the brain, it's a ipsilateral or contralateral.
Yeah. Wait, hold on.
Sorry.
Don't pull too much.
No, we got it. Don't worry.
That's right.
Okay, quick.
Well, I just remember the story that Stevens and when she was working with Netflix, that feels many years ago already.
Yeah.
Connection between electrical activity and
Yeah.
calcium signaling, and
Yes.
yes. If you have any comments,
Yeah. No, we
there's
haven't.
any connection between the two.
We have not really focused on the calcium signaling. So Ethan Hughes is in our group, and Ethan has a student who is really focused on calcium signaling. And they do the cortical window, so they could actually do a lot of the in vivo stuff that we have a harder time. But we have not looked at that. But yeah, I'm sure that's part of this. Yeah.
More questions? There is no more questions. We have a coffee break and we return soon for the symposium. Ahora tenemos dos Zooms. Great.